Welcome to all fours e-learning classes. Now today in this session we are going to discuss about the structure of male gamete, that is nothing but spermatozoa or sperm. Now this is the topic of today. What is this spermatozoa or sperm? The previous class we have discussed about spermatogenesis. Spermatogenesis is the process by which male gametes called as sperms or spermatozoa are released. Now if you take out a single spermatozoan, now from the primary spermatocyte there are four spermatozoa are formed. They are attached to the Sertoli cells and they get discharged into the lumen. This is called as spermiation. Now these sperms which are formed, it shows the following structure. It has the following four parts. The spermatozoa has following four parts: head, neck, middle piece, tail. Now these are the four parts of a male gamete, nothing but a sperm or spermatozoa: head, neck, middle piece, tail. Now here is the spermatozoan, the distinct part, the clearer part, the distinct clearer part with a large amount of cytoplasm with clearly visible in the sperm is that part called as head. The anterior part of the sperm, this is that head. Now head, mostly it is present here. In the head mostly the cytoplasm is completely occupied by the nucleus. A nucleus occupies the entire structure of the head. Now the head also consists of the anterior most part. There is a cap like structure. This is called as acrosome. Now first we will discuss here what are the parts which are there in a sperm or spermatozoa. Now here is head, the first part. This is head. This is anterior most prominent part of the sperm. This is the anterior most prominent part of sperm. Inside this is a head, there is a haploid nucleus. It means head is completely occupied by a haploid nucleus. This is that nucleus. And covered on the head, it is covered by a cap like structure. The head covered by cap like structure. This is called as acrosome. Acrosome. The nucleus has, what is that? Nucleus is having chromatin. A haploid nucleus consists of chromatin, densely packed chromatin with a single, what we are saying, haploid set of chromosome. Single or haploid set of chromosomes. Now this is that part, acrosome, cap like structure on the head. Now this is a derivative of a Golgi complex or modification of a Golgi complex. This acrosome is modified Golgi complex or Golgi body. Now this acrosome, the head consists of cap like structure, acrosome. This acrosome has spermalysins. Now what are the sperm lysins which are there? What are the sperm lysins which are there in the acrosome? Now this is sperm lysins such as the following three. The sperm lysins which are three. One here is granulosa digesting enzyme. This is called as hyaluronidase. Hyaluronidase digests the hyaluronic acid in between the granulosa cells. Now hyaluronidase digest hyaluronic acid between granulosa cells of ovum. This is one spermolysine, one enzyme. Then the other one, corona penetrating enzyme. Corona penetrating enzyme. Now this is one which digests the corona radiata of the ovum. It digests corona radiata of ovum. Then the other structure, the other enzyme, this is acrosin or jonalysin. Acrosin or jonalysin. Now it digests the zona pellucida of the ovum. It digests acrosin or otherwise jonalysin. It digests zona pellucida of ovum. Now these are the three enzymes which are present called as spermolysins. Acrosome, the head, consists of spermolysins which digest the corona radiata as well as hyaluronic acid and zona pellucida. Now these enzymes are hyaluronidase, digest the hyaluronic acid between the granulosa cells of ovum. Then corona penetrating enzyme, it digests the corona radiata of the ovum. Jonalysin or acrosin, it digests the zona pellucida of the ovum. Now these are three are the spermolysins present in the acrosome, that is head region. Now the head region acrosome also consists of certain inhibitors, now which actually get activated. Now this inhibitor will be released and the head get activated in the female reproductive tract. That process is called as capacitation. We will discuss in the later classes. It has some inhibitors on its head. These inhibitors are lost in the female reproductive tract. This is called as capacitation. Okay, this is a head, anterior most part, most prominent part. Cytoplasm is present, which is fully occupied by nucleus. Nucleus is a haploid one set of chromosomes are present. This is haploid and it has covered by a cap like structure called as acrosome. This acrosome is modified Golgi body. This is, now it consists of enzymes, three types of enzymes: hyaluronidase, corona penetrating enzyme, acrosin and jonalysin. Their functions. This is about the head. Next, the head is connected to a structure. What is that structure? Middle piece. The head is connected to middle piece by a indistinct structure called as neck. Now here next, neck. Now this part is the neck which is connected to the middle piece. Now this neck is a indistinct, not clear. It is connected from the head to middle piece, this part. This neck has two that is pairs of centrioles, neck and indistinct part connecting head with middle piece. It has two pairs of centriole. This is anterior centriole, this is proximal. Now posterior centrioles, these are distal. Now here is two pairs of centrioles. Centrioles are basal bodies. Centrioles or basal bodies are present, two pairs. Now this is proximal centriole, distal centrioles. Now here is the centrioles are proximal centrioles, one pair, distal centrioles, another pair, distal centrioles. Now what is the function of this proximal centriole? Now they participate in the spindle formation during the division, that is a zygote division. When the sperm meets ovum, there will be zygote formation. In the zygote formation, spindle formation takes place by these proximal centrioles. Proximal centrioles participate in spindle formation during division, zygotic division. Whereas these are the distal centrioles. They form an axial filament which runs through the middle piece into the tail, forming a flagella of the tail. Distal centrioles, they form axial filaments which run through the middle piece, enter into the tail to form an tail. Now they form an axial filament which runs through middle piece, into the tail, forming the flagella of the tail, which runs through the tail. This is middle piece, forming flagella of tail. Now what we are discussing is neck, which is indistinct, not clear, connecting head to middle piece. It has two centrioles. The two centrioles are proximal centrioles, one pair, distal centrioles, another pair. What is the function of proximal centrioles is they participate in the spindle formation during zygotic division, whereas the distal centrioles, they form axial filaments which runs through the middle piece, enter into the tail to form the flagella of the tail, useful for the movement of the tail, which consists of microtubules, flagellar movement for 9+2 arrangement of microtubules. Now this is a distal centriole. This is about the neck. Next we have middle piece. This is middle piece. This is that middle piece is. Now middle piece is also called as energy chamber. Now it consists of spiral arrangement of mitochondria. Now these are the mitochondria. This is spiral arrangement of mitochondria. Mitochondria are arranged in the form of a spring-like structure. This is what we are saying, spiral arrangement of mitochondria in the form of a sheath. Here the middle piece contain an spiral arrangement of mitochondria in the form of a sheath. Now this is called as Nebenkeron. Now it is energy chamber because it provides energy for the swimming movement of the sperm. Now tail shows the flagellar movement, swimming movement. Now for the movement of this tail, the energy is generated by middle piece, which consists of large number of mitochondria arranged in the form of a spiral. That spiral arrangement of mitochondria in the form of a sheath, this is called as Nebenkeron. And if you observe the cytoplasm here, the cytoplasm which is very less, the small amount of cytoplasm present in the middle piece, it is called as manchet. The cytoplasm is very less and represented in the form of manchet. Represented in the form of manchet. Now this is cytoplasm in the middle piece is called as manchet, which is not clear. Now hence we are saying this is a very less the cytoplasm, whereas a mitochondrial sheath is called as Nebenkeron. This is considered as energy chamber. Now it is also having here, it also has the ring centriole. This is a ring centriole through which what are these structures? Axial filaments from the distal centrioles, they passes through this ring centriole. The function is unknown. Okay. Now this is about the middle piece. Then about a tail. What is this tail? Now tail is the longest part of the sperm. This is the longest part of the sperm. Tail. This is longest part of sperm. Which show flagellar movement. Which show flagellar movement. By taking energy from the middle piece, the tail show flagellar movement. Now this flagellar movement, which is shown by the tail, by which the sperm shows movement. The movement of sperm is due to the presence of tail. For suppose, if any reason tail is absent, it does not show the movement which is shown by the sperm. Hence, this is very important structure, longest part of the sperm. This is. And now it consists of the flagellar part from the distal centrioles. Okay. Now this is tail. Now the entire sperm is covered by plasma membrane. This is that plasma membrane covering the entire sperm. This is that plasma membrane covering the sperm. Now this is the structure of sperm or spermatozoa. The spermatogenesis results in the formation of these structure. Okay. Here, how many sperms are released? Now where they are released? Now a male human being releases, this is that, what is that? Semen. Male human being releases semen. This is semen. Now semen is equal to formed elements plus seminal plasma. Formed elements are nothing but sperms, whereas seminal plasma is the secretion, white milky white color secretion by 3 glands. What are those? Seminal vesicles, prostate gland, as well as bulbourethral gland. Now the seminal plasma is white, milky secretion. This is alkaline secretion, that is nothing but fluid medium is nothing but seminal plasma. Now the sperms along with this fluid medium consist of white milky alkaline secretion produced by seminal vesicles plus prostate gland plus bulbourethral glands. Now these 3 together secrete this seminal plasma. If this seminal plasma is not there, the sperms does not have nutrients as well as movement. Now it means the seminal plasma providing sperms freely moving through and also provides nourishment, nutrients, avoid desiccation, that is the death of the sperm does not take place due to dryness. Now it means providing nourishment, that is nutrients, as well as a medium for the sperms to swim. Okay, this is pH of this semen. The semen pH is 7.4 and this is alkaline, what we are saying, slightly alkaline in nature semen is. Now in a single ejaculate, this is single ejaculate. In a single ejaculator, there will be 2 to 6 ml of semen is released, 2 to 6 ml. On average, it is 4 ml. Now, in a single ejaculate means at one sexual intercourse, this is a single ejaculate, the semen on average, this is 4 ml. Now, this semen consists of 1 ml of semen, 1 ml of semen has 120 millions of sperms. 1 ml of semen, it has 20 to 120 millions of sperms. It means average we are saying 300 to 400 million sperms are released. How many? This is 300 to 400 million of sperms are released per a single ejaculate. 1 ml of semen consists of 20 to 120 millions of sperms. Now this is 300 to 400 millions of sperms are released. It means their number is very, very high. 300 to 400 millions of sperms are released in a single ejaculate. Okay. Now if you observe, there should be at least 60% of sperms must have a morphology, that is size and structure, proper morphology. Now this is very important for fertilization. Now here what we are saying, this is this is what we are saying, for proper fertilization at least 60% of sperms must have definite size, shape. This is proper morphology. then at least 40% of sperms must show vigorous motility. Now this is what we require for fertilization. At least 60% of sperms must have definite size and shape. This is morphologically, they should have this size and shape. And at least 40% of the sperms must have a vigorous motility. Vigorous motility means not normal motility, very fast. Very fast, they should be moving very fast. When they move very fast, then only fertilization can occur. It depends upon. It means 60% should have normal size and shape, as well as 40% of sperms must have vigorous motility, not normal motility. Vigorous motility means very fast movement it should show. Okay. Now for suppose if there is less than 20 million sperms are there, if less than 20 million sperms, now this condition is called as oligospermia. Oligospermia means a decreased count of sperms. It may also lead to sterility. This is less than 20 million are present for ejaculate. Now this condition is called as oligo. Oligo means very few. If sperms are absent or nearly absent means hundreds or thousands for suppose. Now this condition, if sperms are absent, this condition is called as azoospermia. Azoospermia means absence of sperms in the semen. Now vasa recta, for suppose is cut or seminiferous tubules are blocked, the sperms may not be observed in the semen. Seminal fluid may be released, but sperms are not observed. When we cut vas deferens, if we cut, there is no sperms in the semen. Even seminiferous tubules are blocked, there will be no sperms in the semen. Okay. Now this is sperms, absence of sperms is called as azoospermia. Similarly, if there is no motility, if motility is absent, if motility is absent, it means sperms are unable to move, sperms are not showing any movement. Now this is called as ethnospermia. It means sperms are unable to move in the seminal fluid. Now this may also lead to sterility. This is problems related to infertility problems. This condition and this condition. Now this condition does not, they are present but less in number. But azoospermia or ethnospermia, it results in the problems related to infertility. Okay. Now this is the structure of male gamete or spermatozoa or sperm. Nearly 50 micrometers in size, not visible to our naked eye. Whereas ovum is visible to our naked eye, sperms are not visible. 50 micrometers in size. Okay. Now this is about the structure of male gamete or spermatozoa. Thank you.